If you remember, last time we have discussed about the protein synthesis first part. Okay. Do you remember this diagram where only first part, that is transcription, we have done, we have completed, where transcription is the first step in the protein synthesis, and transcription is nothing but the information from the DNA. It is carried in the form of code language to mRNA, right? And this it takes place in the ready to function, that is considered to be non-functional RNA or mRNA. It has to be processed, and in the processing, what we studied, the three steps, that is, one is splicing, that means removing of non-coding sections or nucleotides. That means these are called as introns. Introns are removed, which are non-coding regions, and exons only are kept. So with the help of these spliceosomes, these introns are removed. This is the one step that is called as splicing. Then addition of phosphates at the 5' end, that is methylated methylene triphosphate at 5' end, that is called as capping. And telling is what? Addition of adenine nucleotides at 3' end, that is called as polyadenylation, and this is what called as capping. So these three steps when are completed, then this mRNA is ready to work, that means function, that has become mature now for the next step. And what is the next step? As I told you, is translation. But once it is ready, then only through the nuclear pore it will move to the cytoplasm, to the actual site of protein synthesis. What is the site of protein synthesis? That is
Madam, shall I
you might
interrupt?
have forgotten. Last year we have learned it. Yes, ma'am.
Your voice is cracking, huh? Just see that.
Okay, my voice is cracking.
Yeah.
Just a minute. Now lights are. Madam, can you hear me?
Yes,
Hello.
yes.
I will try, but if you can't hear immediately, let me know.
Yes,
Okay.
sure.
Now these are the types of RNAs. First is mRNA, which is single-stranded structure with the nucleotides on it, and this is 5' end, and this is 3' end. Simple single-stranded RNA, that is called as messenger RNA. RNA or messenger RNA, and the word itself suggests that it is carrying message in the form of code language, that means genetic code on it from the DNA for required for the process of protein synthesis. Now this is the structure of rRNA, okay, where this is rRNA and this is the ribosome. I will tell you the structure of ribosome again, but this is the rRNA who directs the mRNA to the site of protein synthesis. And this is the transfer RNA, where it has two unequal arms. This is the short arm which ends with 5' end. This is the long arm which ends with the 3' end. This is also called as amino acid acceptor arm, because amino acid required for the protein synthesis gets attached to this long arm which ends with 3' end. And opposite to this unequal arm, whatever this region is called, this loop is there, that is called as anticodon loop. That means whatever the nucleotides present on this loop, they are exactly complementary, should be complementary to the codons on mRNA, which is required for the process of protein synthesis. Now, see, this is the structure of our ribosomes. And before we come to the translation part, you should know the structure of ribosome. This is this in case of eukaryotes, the ribosomes in both the cases, it is made up of two subunits. One is larger unit, the other is smaller unit. This larger subunit in case of eukaryotes is of 60S subunit, and the smaller is of 40S. Sweatberg's units, that is called as. So 40S, that is called as 40S subunits. And in the cytoplasm, these are in the dissociated form, but at the time of protein synthesis only they come close to each other. They come in contact with each other. Now we'll start with this one more structure, that is the called, this is called as tRNA, the adapter molecule. Adapter, why? I'll tell you now. See, this is the short arm which ends with 5' end. This is the 3' end which carries or accepts amino acid required for the process of protein synthesis. This is the structure I'm showing you. This is the mRNA, okay, with 5' end and 3' end. First nucleotide, whatever is these all nucleotides, 3 consecutive nucleotides, they form a single codon, and this is the opposite end of this anticodon, that is called as anticodon loop. The codons or the nucleotides which are present on the anticodon loop, they should be exactly complementary to the codons on the mRNA. Only that tRNA will be able to come in contact with the codons on mRNA which are having the complementary nucleotides. Okay, so this is called as in anticodon loop. Now we'll start with the actual process of protein synthesis, that is the second stage, second step of protein synthesis. This is called as translation. Now everybody knows the meaning of translation. Translation means what? Whatever the information is there carried on mRNA from DNA is to be translated, and that work, that is what called as translation, because that information is in the coded language, that is in the form of genetic codes, and that is to be translated in a specific way. That means these amino acids which are carried by this tRNA, they are in a specific sequence to form a polypeptide chain. So the process of translation requires, now what are the requirements for the translation, that we'll see first. See, these are the requirements. What it requires: mRNA, as I showed you, which carries codons, that means genetic language is in the form of code, that code language is in the form of codons. Then tRNA, which carries the amino acids, nothing but it is the polynucleotide chain. Poly means many, nucleotides, they are carried, they are in a specific sequence. What are these nucleotides? Amino acids. They are, proteins are made up of amino acids, and these amino acids, they are carried by tRNA. Ribosomes, which is considered to be the site of protein synthesis. Then ATP. Why ATP is required? Because as you know that for any process, energy is required, and this energy is taken in the form of ATP. So these amino acids are to be activated, and for that this ATP is required. Then Mg ions. Why they are required? Because they help to make the two subunits of ribosomes to come close to each other during the process of protein synthesis. Then enzymes, different types of enzymes are required. At every time, as and when the name of the enzyme comes, I'll tell you the role of the enzyme. Then elongation means these nucleotides, when the amino acids are coming close to each other, carried by the tRNA, they are to be joined, and that is what elongation takes place. And the movement of these ribosomal subunits along with the codons on the mRNA, that process is called as translocation and the release factor. I will tell you these 2 points at the end. Amino acids form raw material for protein synthesis. That means what? Proteins are made up of amino acids, and everything is decided by DNA. As I told you last time, DNA only decides the which type of amino acid is to be produced. So the number of which type of protein is to be synthesized. So the number, sequence of amino acids is the DNA itself. Now you know there are 20 different types of amino acids present which form proteins, and these are available in the cytoplasm. Now as I told last time that, and right now also, that mRNA is produced in nucleus, and through linear pores now functionally it comes to the cytoplasm, to the actual site of protein synthesis. DNA
I'm
controls
sorry,
synthesis of proteins.
madam. I'm very
Yes,
sorry to interrupt you.
yes,
There is
ma'am.
someone else who is also presenting. He has left the meeting. You can continue.
Okay,
Okay,
okay.
now you can continue. He has left.
But voice is audible, na, madam?
Yes, no problem. I'm sorry. Today I had to interrupt you, but you can continue now.
Yo, it's okay,
Yeah,
it's okay.
yeah.
Yeah, yeah, yeah. DNA controls synthesis of proteins having amino acids in specific sequence, and this control is possible through transcription of mRNA, which I already discussed. RNAs serve as intermediate molecules between DNA and proteins. You know the steps: DNA to RNA, RNA to protein. This is the sequence of protein synthesis. That means information from DNA is carried to RNA, and RNA from that RNA actually in the synthesis of protein. So this is the sequence. So RNA is considered as intermediate molecule. Ribosomes serve as site for protein synthesis. Each ribosome consists of large and small subunits. These subunits occur separately in cytoplasm. Only during protein synthesis, these two subunits get associated together due to Mg ions. Just now I told, isn't it? So a ribosome has one binding site for mRNA and three binding sites for tRNA. They are P site, A site and E site. I will explain when the diagram will come. Okay. A mechanism of translation, that means synthesis of polypeptide chain, which involves these three steps. First is initiation, second is elongation, and the third is termination. Remember, now I will revise it in the form of video, but right now you try to understand. Okay. Initiation is the first step, elongation next second step, and termination. Initiation means start. How the translation starts? For that, very important thing is activation of amino acids is to be done. And how these amino acids are to be activated? By utilizing energy in the form of ATP molecules. In case of DNA replication also, you know that these nucleotides are to be charged, and where ATP is utilized. The same way amino acids are to be activated, energized, and that is done with the help of ATP. And when this amino acid binds to that binding site of tRNA, then it forms tRNA amino acid complex. I will show in the diagram, and I showed also how the at 3' end that amino acid is getting attached. 3' end of tRNA, this activated amino acid gets attached, and that tRNA with the activated amino acid is called as tRNA amino acid complex. A small subunit of ribosome binds to the mRNA at 5' end. So after this tRNA complex is ready, what happens? This small subunit of ribosome binds to the 5' end of mRNA. mRNA is already has come to the ribosome. Ribosome means in the cytoplasm where the actual process of protein synthesis takes place. That means ribosome. So ribosome binds 5' end of mRNA. Then tRNA, which is charged. That means what? tRNA amino acid complex that comes in contact with this mRNA. That is the first codon now. So initiator codon is always AUG present on mRNA, which initiates the process of translation. That means what? You always remember. Here I will show you. This is the mRNA, where you can see these all. This is the 5' end. This is the 3' end of mRNA. This is AUG. AUG is always considered to be initiation codon on mRNA, or this is also called as start codon on mRNA, which initiates the process of protein synthesis. That means translation. Now this is tRNA. This is the anticodon loop of tRNA, and as I told you, these anticodons which are present, these nucleotides are exactly complementary to the codons on the nucleotides on, that means codons on the mRNA. So see, and this large arm which ends with 3' end, here it carries the amino acid required for the protein synthesis, and it is always methionine in case of eukaryotes and formylmethionine in case of prokaryotes. So this is carried, this amino acid is attached to the 3' end of this tRNA, and anticodon loop, it carries the nucleotides as UAC, which are complementary to the codons on mRNA. That is the first codon, that means start or initiation codon on mRNA at 5' end. So you can understand A is complementary to U is complementary to A, and G is complementary to C. So this is the sequence of anticodon. Okay, so this is first codon, this is second codon, and this is the last end, that is the 3' end. I'll come to that point later. So, and these are the charged or activated amino acids. You can see alanine, trypsin, like that, glycine. So these are the amino acids carried. These are the tRNAs carrying amino acids required for the protein synthesis. And when the amino acids are attached to the 3' end, this is considered to be amino acid tRNA complex. So such type of amino acids will come. So here is the first step where AUG is the first codon, starting codon on mRNA, and tRNA with a amino acid methionine comes in contact with the complementary nucleotides on it at anticodon site, and the bihydrogen bonds in between, they join. Okay. Now, this is what first step, that is called as initiation. Now, second is what? Elongation. Elongation means what? The second amino acid, this tRNA, amino acid tRNA complex will come and binds to the second codon on mRNA. Then third will come, binds to the third. But this process of elongation, how it works, that I will show you in the next diagram. Now, see, this is the initiator tRNA binds with the initiation codon AUG by its anticodon UAC through hydrogen bonds. It carries activated amino acid methionine in eukaryotes and formyl methionine in prokaryotes. Now, the large subunit, once this tRNA gets attached to the mRNA by hydrogen bonds, then the larger subunit of the ribosome comes and joins to the smaller subunit, which requires MJ ions. MJ ions are already there in the cytoplasm, which helps to join this large subunit to the smaller subunit. And then initiator charged tRNA with activated amino acid methionine occupies the P site of ribosome, and A site is vacant. Now I will show this what is A site, P site in this diagram. Okay. Now you can see this diagram. So this is the mRNA. Can you see this red color structure? This is mRNA carrying the codons on it. This is the tRNA. This is the amino acid which the tRNA is carrying, forming the amino acid tRNA complex. And this is the initiator tRNA which carries methionine. And this first one carries the AUG. First starting codon, initiator codon on mRNA is AUG. This is the smaller subunit. Okay. So first of all, this mRNA joins to the smaller subunit. Then this tRNA which carries amino acid, that means amino acid tRNA complex comes in contact with mRNA on P site. Now what is P site? And then this larger subunit joins. Now I will tell you what is the P site and what is the A site. See, can you see these three compartments? This is E site, this is P site, and this is A site. Very simple to remember. See, A site means what? Amino acid acceptor site. So that's why this is called as A site. P site means what? Peptidyl site. Means this peptide bond is found. What is peptide bond? I will tell you in the process of elongation now. And this is E site means exit site, from where this tRNA gets out of this after its function is over. That is what exit point or exit site. Now, what happens in the elongation? I will show you in this. Now, this is the first, see, this is the first tRNA carrying the amino acid. Okay, it gets entry from A site and it moves forward to the P site. Okay, this is the P site. Why P site? I'll tell you. This is the first P peptidyl site. Then first it gets entry here, enters from A site, then it occupies the next site, that is the P site, so that A site is made free or vacant to come for the next amino acid to come, or next tRNA carrying the amino acid to come and join. So this is the second tRNA. It will get entry through A site on this, where this tRNA is also carrying the second amino acid, that is the activated amino acid, and it has the anticodon loop which is complementary to the codons on mRNA. So this is P site, this is A site. Now how we will call it P site? Now see, once the second amino acid comes and joins and occupies the P site, what happens? This tRNA carrying first amino acid, it forms a bond between the second amino acid, that is called as peptide bond or peptide linkage. Okay, so peptide bond is formed between first amino acid and second amino acid, and then once this peptide bond is formed, once this amino acid gets attached to the second amino acid on second tRNA, this tRNA is free now. So this tRNA will get out of this, that is exit, through exit site, it will come out of this side. So this is called as exit site. So like that, coming, entering the train bogie through this entrance, that is A, and then occupying this passenger as a seat, and then when the destination comes, coming out of the door, this is the exit door. So you can just remember, imagine like that, that this is the bogies, that these TRNAs are the passengers entering through the entrance, that is A. Then this is the seat where P passengers are occupying the seat, and when the destination comes to through the exit door, they are going out, and this is the track. You can consider the mRNA as a track, and this as the bogie and all. I will tell you again this. Just try to understand that second tRNA is here. Now, when this side, that means P side is free. Now, what will happen? This tRNA, which is on P side, will move to the A side. Okay, now this tRNA will be moving to the P side. That means what? It is moving means translocating. This is what called as translocate. Trans means change. As you know that trans is any change in the position, in the location, whatever. Changing phase is considered to be trans, right? It is location. So location is changed. From A side, it is moving to P side. That means that is what called as translocation. When the second RNA, first RNA is getting exited, it is moving out of this because its work is over, because it has its amino acid is joined to the second one. So this place is vacant now. So the second tRNA will occupy the P side, so that the third tRNA will come, and the third tRNA will come through A side, that means entry side, that means amino acid acceptor side. So this is what called as translocation, that is the moving of the translocation of these tRNA or the codons on along with the ribosomal subunits. This is what called as translocation movement, and the peptide bond is formed between the first amino acid and the second amino acid. Then the third amino acid will come through the A side, that is the A site of on this ribosome, and then again what will happen? This will move further. So this will be continued till what? See, mutation. I will explain this part again. During this process, activated amino acids are added one by one to the first amino acid, that is methionine. Addition of amino acid occurs in 3-step cycle. Codon recognition, as I told, amino acid tRNA molecule enters the ribosome at A site. Anticodon binds with the codon by hydrogen bond. Then first amino acid tRNA molecule moves to the P side, and second amino acid tRNA is attached to the second codon on mRNA at A side. And those two amino acids join by peptide bond. Now see here, what is the enzyme used? That is ribozyme. And then which first amino acid will be moving to the next side, that is E side, through exit side, it will get out of the that site. Okay, it will come out of, it will be kicked off at E site, that is the exit site. Now what will happen now? Next, that is translocation. As I said, the tRNA at A site carrying a dipeptide. Now this is called dipeptide. Why dipeptide? Di means two peptide bonds are formed, so that's why called as dipeptide. A site moves to the P site. This process is called as translocation. In translocation, both the subunits move along in relation to tRNA and mRNA. tRNA is also moving like a passenger coming inside and getting out of this one, that is the site, that location, that means mRNA. Then mRNA is also moving. During this process, first uncharged tRNA is discharged from E site because it has that amino acid which it was carrying is joined to the second RNA by peptide bond. So this process is repeated as amino acids are added to polypeptide. And the how till what it will happen, till when? That is till the last amino acid on the mRNA, that where it will not recognize any of the codon. That is what called as termination of the reaction or termination of the polypeptide chain. That means here up to that point the entire polypeptide chain is formed. So at the end of mRNA there is a stop codon called as UAA, UAG or UGAE. Out of these three, any of the codons, if it is there at the end, then it will not recognize any of the amino acid. That means there is the completion of polypeptide chain. it is exposed at A site. It is not read that if there is UAA or UAG or UGA on this mRNA, it will not read and will not be joined. So anticodon of the tRNA will not be joined to the mRNA, codon on the mRNA. So these are called as stop codons. Now, so see in this diagram, see this is the exit site. This is the polypeptide chain, because whatever the amino acid you can see here, this is amino acid 1, 2, 3, 4 like that, and in between the two amino acid, whatever the bond you can see, this is called as peptide bond. Poly means many and peptide bonds are formed, and so polypeptide chain this is called as. And when it will reach the last codon, suppose this is the last codon, so this is the last codon which will not recognize any of the anticodon on tRNA. So in that case, what will happen? Here will be the termination of the reaction. That means polypeptide chain is formed, and this is what stop codon or termination codon. And after that, what will happen? Everything, this polypeptide chain is formed, it will get released, all the tRNA will be released from this side. These subunits also get will be dissociated. mRNA's role is also over. So the release factor binds to the stop codon and thereby terminating the translation process. The polypeptide is now released in the cytoplasm. And then once the polypeptide chain is formed, that is released in the cytoplasm. And then two subunits of the ribosome also get dissociated from each other. And the last tRNA will be released in the cytoplasm. Then mRNA also, its role is over. So mRNA also has some additional sequences. Remember, this one is additional point. You can remember. It is having additional sequences at the end point. That means as 3' also and 5' end also. These are called as untranslated regions. These are considered as untranslated regions. That means UTR. So these UTR are present at 5' end also. That means start before start codon. That means AUG, before AUG also, and after 3' end also. So they are required for the efficient translation process. Okay, so this is what I just want to show you the video which I will explain you. It will take little time. Okay, before I start, Lele madam, can you hear me?
Yes,
Uh,
yes, I can.
any of you? Ha ha. Now I am
But it's
starting the
cracking.
video.
It's cracking
Just
in between, huh?
this.
Your voice is cracking in between.
Okay, okay. Yeah, because this is going on data, no? That's why.
Okay.
Okay, but everything was audible, na? And this one, you were able to see, na?
Yes. Yes. Able,
Now
able
I
to.
will start the video. Yes, I will start the video. The voice of the video you will not be able to hear, but I will explain as and when required. You just very carefully see this video, which explains transcription as well as translation. See, this is the DNA. You can see the promoter site, terminator site. These are the genes, double stranded structure of DNA. See, two parts are, one is transcription, which takes place inside in the nucleus, and the second takes place inside the cytoplasm. See, this is double stranded DNA and this is the RNA produced. And in translation, These are the sites, E site, P site, and A site. See, this is what? This is polypeptide chain. Now, transcription. This is the template DNA. RNA polymerase gets attached. So, initiation, elongation, and termination. These are the three stages. Promoter region is the recognition site for RNA polymerase. It gets attached to the promoter site, and then this is RNA polymerase. Double helical strand gets unwinded. One of the strand works as template or parental strand. The addition of complementary nucleotides takes place, and this is the RNA which is produced. It gets dissociated from the template RNA. Once the polymerase reach the stop codon or the terminator region, this RNA polymerase is released and the DNA strand is dissociated. They dissociate from each other. This mRNA contains non-coding and coding regions. Exons are coding regions and non-coding regions are called as introns. This is to be processed because this is immature RNA. So non-coding regions are to be removed. This is 5' end. This is 3' end. Now splicing is done with the help of proteins. Proteins and RNA complex called as spliceosome, which removes non-coding regions called as introns. And only exons, they are joined to each other by ligases. And then by capping and telling, it comes out of the nucleus. So this is the mRNA. Now translation, ready for translation. These are the nitrogenous bases. 3 continuous consecutive nitrogen bases called as codons. There are total 64 codons which code for 20 different types of amino acids. First codon is called as start codon or initiation codon. First, this messenger RNA binds to the small unit of ribosome. Then amino acid which is attached to the 3' end with the anticodon site with the complementary nucleotides to that mRNA on mRNA codon on mRNA comes. So this is what complementary A, U, A and GC. These are codons on mRNA and these are the anticodons on tRNA. This is the initiation. Then larger subunit binds to this smaller subunit. This is P site. This is E site. And this is A site. These are the E region, P region, A site or A region. During elongation, amino acids are brought from A site by transfer RNA molecule, and the anticodons and codons should be complementary. That tRNA only will be able to come in contact. So it is very specific. The charge transfer RNA comes to the A site. Their peptide bond is formed between the first and second amino acid at the P site. Now this P site RNA moves by one codon to E site, and then it is now uncharged, which gets exited through E site. Then the third amino acid comes, and such type of chain of polypeptide nucleotides takes place. So this is a polypeptide chain, and this is E site, exit site, and release factor binds to the A site, which do not code for any of the amino acid. Then both the subunits associated, and this is what polypeptide chain. Okay. Now I'll come to the again our part. So this is the end. This is what translation. Now, the next part of this is, I hope you have understood with the help of this video. And the next part which we are going to begin is with the genetic code. Now, just now I use the words as codons and all. Okay. So in short, now you'll come to know what exactly these codes are. Okay. So we are going to study this under the heading as genetic code. Genetic code is what? DNA is a master molecule of a cell that initiates, guides, regulates, controls the process of protein synthesis. It carries the requisite information for the synthesis of proteins. About 20 different types of amino acids are involved in the process of synthesis of proteins. DNA molecule has four types of nitrogen bases to identify these 20 different types of amino acids. Okay. So question arises then, how is it possible that 20 types of amino acids are encoded by four types of nitrogen bases? Isn't it just I showed you while showing the protein synthesis translation part, transcription and translation video, you observe the chart of 64 different types of amino acids, isn't it? So that time you observed, right? What you observed, there are total 64 codons. Now, you know that there are four different types of nitrogen bases, so four different types of nucleotides are present, right? If only one nitrogen base or only one nucleotide, if it codes a single codon, if you consider it as a single codon, then how many total? Only four total amino acids will be coded, right? But how many are there? Total 20 different types of amino acids are present naturally, isn't it? So then if it is two nitrogen bases, if codes for a single amino acid, then what will be the result? 4 raised to 2, total 60 amino acids only will be produced, isn't it? But there are 20. So if then three nitrogen bases code for a single amino acid, that means three nitrogen bases, they form a single codon which specifies a single amino acid. Then only what happens? Then only there are chances of more. That means total what? 4 into 4 into 4. How many total? 64. Means 64 codons are available. That means it is, we can consider that yes, 20 amino acids, for 20 amino acids less number is not useful, so more number 64 codons are available. That means one amino acid might be recognized by more than one codon. Okay. Now, what is genetic code? In short, I'll tell you first. Genetic code is the sequence of nitrogen bases in mRNA molecule which encloses information for the synthesis of protein molecules. The genetic code is the set of rules by which information encoded in genetic material, that is DNA or RNA sequence, is translated into protein by living cells. Just now you observed in the, so that's why I explained the translation in continuation with the transcription, because that is a continuous process. That's why I'm taking this part later, little bit later. I just told you that whatever the information is present on DNA, that is taken in the code language on mRNA, and that is code language is in the form of codon. So codons are present on mRNA, and that is nothing but the genetic codes. These are in the form of genetic codes. And how it is considered, just now I showed with the help of four types of nucleotides. What is the discovery? How it took place? To understand how proteins are encoded began after the structure of DNA was discovered by James Watson and Francis Crick, who first of all proposed the double-stranded helical model of DNA, isn't it? So first elucidation of codon was done by Marshall Nirenberg and Henry Mattei in 1961 at National Institute of Health. Now, genetic code is a dictionary, consists of genetic words called as codons. As I showed you, codon 1, codon 2, codon 3, like that. So Each codon consists of 3 bases. So that is called as triplet. Try to understand these terminologies used in the genetic code. As I showed you AUG, that is what the first A, U and G nucleotides. These 3 nitrogen bases together forms a single codon, right? That means what? A single codon, what it contains? Triplet. So that's why this is called as triplet. So each codon consists of 3 bases consecutive. There are total 64 codons, as I showed you, but only 61 codons they are able to recognize 20 different types of amino acids found in proteins. Okay, 3 codons do not code. And what are these 3 codons? They are called as stop codons or termination codons. Now, few characteristics of genetic code we are going to discuss. As I said, first is what? It is triplet. That means 3 nitrogen bases form a single codon. Okay. Then one more characteristic feature is it is universal. Universal means what? All living organisms share the same code. We can read the DNA of a bacterium, a plant, a worm, or any type of animal using the same code. So this is what considered to be proof for evolution. That means if AUG is methionine in myself, it will be same in plants, it will be same in bacterium, in same in all type of animals like that. That's why this is called as it is universal. Then second is degenerate. Degenerate means what? As I told you in the first this thing only, how it is formed. If only 4 nitrogen bases, one nitrogen base if forms one codon, then only 4 codons will be there. But there are 20 amino acids available, so it is not possible. So if 2 nitrogen bases they form, then only 16 amino acids, 16 codons will be available, which are with the number is not enough for 20. So 4 into 4 into 4, that means 64 codons. If the 3 nitrogen bases come close to form a single amino acid, the codon, that means these amino acids can be, these codons will be able to identify 20 different types of amino acids. So that means what? Each amino acid has more than one codon. Sometimes there are 2 codons, sometimes there are 3 codons, which are specifying the same amino acid. That is what called as degeneracy of the genetic code. This is one of the important characteristic features of genetic code, that it is degenerate. So first is universal, second is it is triplet form, as 3 nitrogen bases form the single codon. Then the third is what? It is degenerate. Then one more thing related to that you need to remember, just now I mentioned there are different types of codons. Some are sense codons, some are signal codons. Sense codons means what? They are involved in the identification of amino acids. That means they code for amino acids. They are called as sense codons. And signal codons means what? They either signal for the protein synthesis to start or stop. That means to initiate or to stop. They are called as terminating codons or initiating codons. So initiating codon, that is AUG, and termination codon is what? There are 3 terminating codons or termination codons. They are UAA, UAG, and UGA. They are also called as umber, orchard, and opal. These are the names of these 3, which stop the reaction. Which do not identify any of the amino acids. So they are called as termination codons. Okay. So in case of eukaryotes, first initiation codon is always methionine, and in prokaryotes, it is formyl methionine. Now, the next one more thing you just have to remember in case of codon and anticodon, that codon is a part of DNA. Okay, so AUG is codon, which will be represented always in 5 prime to 3 prime direction. A will be at 5 prime end, it will start, then the process will be towards 3 prime end. Then anticodon is a part of tRNA, and so it will always start with 3 prime end. This is what the fact you have to remember. Now, what are some of the properties of genetic code? First is genetic code is universal. As I said, genetic code is triplet code. That means 3 nitrogen bases form a single codon. So that is a triplet code. This genetic code is non-overlapping. Non-overlapping means what? Suppose first is AUG, second is CGA. Okay. Then AUG, after AUG, you will not start from GCA or like that. They are not overlapping. Okay. They are not overlapping at all. After AUG, whatever the next comes, that is suppose CGA, then CGA you should read. You should not read GCA or something like that. So they are not overlapping. So the code is non-overlapping. Then one more thing you have to remember, that there is no comma or any type of punctual mark used in the genetic code. They are without any markings or any punctuations used. When you read in continuation, AUG, CGA, CAG, GCA, GCU, like that, 333, like triplets, you should read. You should not overlap, and there is no comma in between. So that is also called as genetic code is commaless. That is non-overlapping. Then the code is non-ambiguous. There is no ambiguity because each code specifies only a specific amino acid only. The same code will not, that genetic code will not code for another amino acid. If AUG specifies methionine, this code will not recognize any other amino acid. That's why this is called as non-ambiguous nature of the genetic code. Now the code has polarity, as I said and showed also, that code how it is read, that is from 5 prime to 3 prime direction. Genetic code will be read from 5 prime, that if it is AUG at 5 prime end, that is what that is at 5 prime end, and it will end at 3 prime end. So this is what called as code has polarity. And the code is degenerate, that means more than one amino acid can be recognized, more than one amino acid can be recognized by more than one codon. That is what called as degeneracy of the genetic code. Okay, so these are some of the important characteristic features of the genetic code. Now we'll come to the next part, little small part. Okay, uh, this is the related to the mutations and genetic code. Small part I will take from this. Mutation means what? It is a genetic change. That is a sudden change in the genetic makeup. That means what? In the DNA sequence. It results in the change of genotype. That means character is changed. If there is change in the sequence of the nucleotides on DNA, that will change its character. And along with recombination, mutation is also considered to be the raw material for the process of evolution, as it results in variation. If there is change, that means there is variation. So obviously, variation always leads to evolution. So not only recombination, but mutation also is responsible or considered as a raw material for the process of evolution, which involves the variation. Mutation involves deletion or insertion or duplication of the segment of DNA. Now, what is insertion and what is duplication, I will tell you. But let me tell you this first. And that results in alteration in the chromosomes. It can also occur due to change in a single base pair of DNA, known as point mutation. Example is sickle cell anemia. And deletion or insertion of base pairs of DNA causes frame shift mutations. See, point mutation means what? If I'm reading the nucleotides on mRNA as if the sequence of DNA, nucleotides on DNA is A, U, G, C, A, like that. If there is change at one nucleotide, instead of A, U, G, C, A, G, if A is missing somewhere or G is missing, it is removed, it is deleted, then what is going to happen? Obviously, the sequence will be changed. Say A, U, G, C, A if it is there, and then C is there. If it is not A, directly C will come. So in that case, because of a single change in the nucleotide, single change, that is called as point mutation. Because of a single base pair change, that is called, whatever the changes take place, that is called as point mutation. How it is sickle cell anemia? You have learned the sickle cell anemia last year, I think you have learned, but that when it will come in detail in the next part, that time I'll explain it more. Okay. And what is frame shift mutation? If the entire set of nucleotides is deleted or inserted, if addition AUG, CGA, like that it is there, if CGA is added or CGA is removed, whichever the nucleotide, if it is added or new nucleotides are added, that is what called as insertion. If the nucleotides are removed from the sequence, that is called as deletion. So the entire set of nucleotide, when it is removed or added, that means if it is deleted or inserted, that is what called as frame shift mutation. So insertion or deletion of one or two bases changes the reading frame from the point of insertion or deletion. So insertion or deletion of three or multiples of three bases results in insertion or deletion of the amino acids regarding from the sequence. Now here we have to compulsory have this question answer session. So let presentation. And stop the recording also. Madam, you can stop the recording and